Welcome to the last lecture of the course Introduction to Complex Biological Systems. So, in this lecture, I will give you a brief description of whatever we have seen so far with an emphasis on some landmark experiments. So, we have learnt these concepts as if that these are known truths, right? But there has been a lot of work that has been done with some really smart people over the years to arrive to these truths that we now take them for granted. So I will just pick up some of the important experiments which we refer to as landmark experiments and discuss them. Some of these you have already seen throughout the course of this lecture, and some will be new for you. So for example, I will start with identification of DNA as a genetic material, and I will discuss these three experiments. Then I will talk about the discovery of the structure of DNA, which is the double helix structure of DNA, and the contribution of all these important scientists in that discovery. Then the discovery of semi-conservative replication, discovery of codon, that when we translate from RNA to protein, we have to go from a 4-base language to a 20-base language, right? So that is where codons become so important. Then discovery of tRNA as adapter molecules, deciphering of the genetic code, which is the dictionary that is used to synthesize proteins from the message that is encoded in DNA or RNA, and finally discovery of RNA splicing. So again, this does not cover all the important experiments that are there in biology, but this will give you a flavor of how real science is done. Okay, so let's start with molecular biology, because all the experiments that I am going to discuss today come under the purview of molecular biology. So what is molecular biology? Here we are talking about important molecules or biological molecules which play important part in these biological systems. So molecular biology is a branch of biology that focuses on understanding biological activities at the molecular level, particularly the interaction between DNA, RNA and proteins. So these are the three very important biomolecules, and we have discussed about them at length in this course. So molecular biology primarily studies nucleic acids, which are DNA and RNA, and proteins and their roles in gene expression, regulation and cellular function. So the flow of genetic information follows central dogma. We have discussed that at length throughout this course, and I will again put the central dogma as the central theme of today's lecture, where DNA is transcribed into RNA, which is then translated into protein, and proteins are the ones which perform all the cellular functions. To do molecular biology, the tools that we need, people have designed all sorts of very interesting and important experiments throughout the years, in the past 100 years. So there are techniques like PCR, DNA sequencing, recombinant DNA technology is something that is very important, gene editing, for example, CRISPR, etc. So molecular biology has revolutionized genetics, medicine and biotechnology, right? So it has enabled us to make some real advancement in techniques like gene therapy, for development of new drugs, even in forensic science and personalized medicine. So this is something that is going to be really big in the next decade that we are going to see. So major advancement in medicinal science, you will see, will be in personalized medicine. So let's focus on this central dogma of molecular biology, because this will be the central theme of today's lecture. So what we have seen so far, DNA is the one which encodes information in most organisms. When that information is copied, that is replication, it is done by DNA polymerase. That information is copied into RNA. So part of the DNA is copied into an RNA, and a reverse process can also happen, which is reverse transcription. So this process is transcription and the reverse process is reverse transcription. So the synthesis of RNA from DNA is done by RNA polymerase, and synthesis of DNA from RNA is done by reverse transcriptase, right? Now RNA can also be synthesized using RNA as a template. So in this case, it becomes RNA dependent RNA transcription, and it is done by RNA dependent RNA polymerase. So this is also something that is found in viruses. Now RNA is used as the messenger from which proteins are synthesized, and this last process is called translation, and it is done by ribosome. and we have seen that for this, the information that is encoded in RNA is converted into the amino acid sequence by using of a table called the genetic code, where 3 consecutive nucleotides code for one amino acid, and that interpretation is done by certain adapter molecules called the tRNA. So, we will talk about the discovery of tRNA and codons in today's lecture. So, the first one is the identification of DNA as the genetic material. So, till this time, all these 3 molecules were known that there are 2 types of nucleic acids, DNA and RNA, and there is this protein, but it was not clear that which of these molecules is the major genetic material, right? And first clue came from this landmark experiment that was done by Frederick Griffith. So, this was discussed in the first week's lecture. So, I am briefly going to talk about it. Griffith was a British bacteriologist. So, in 1928 he performed this experiment which revealed this transforming principle, right? So, what is transforming principle? To do that, Griffith used Streptococcus pneumoniae. So, it is a bacteria, okay, and this bacteria comes in 2 strains, the S strain, S stands for smooth, and the R strain, R stands for rough. Now, the smooth strain is the one which is infectious, so we call it virulent, and the rough strain is not infectious, so it is non-virulent. And the major difference of these two is that the S strain had a protective polysaccharide layer on top, right? So, that why under microscope it looks shiny, it looks smooth, but the rough strain, since it does not have this polysaccharide layer, it looks bit sort of, it will not reflect light, it is not shiny, so it is rough, right? And because of this polysaccharide, it can escape our immune system and actually can cause virulence. So, what Griffith observed that when he injected the S strain into mice, they died, but when he injected the rough strain into mice, they survived. However, the interesting experiment was that when he killed the S strain, so you just boil this bacteria, so it will lyse, everything comes out in the open, and if that solution is injected in mice, the mice survived, okay, because we have heat killed the bacteria. But if you mix this solution with live R strain, incubate it for some time, and then inject that into mice, the mice died, right? And when he looked into the blood of these dead mice, he actually saw that it contains live S strain. So, somehow the R strain got converted into the S strain, right? And that is what is this transforming principle. So, it means that when he heat killed the S strain, the molecules that came out, one of those molecules is able to convert or transform the R strain into the S strain. So, this is the transforming principle, and then people started looking for the molecule which is responsible for this transformation of the rough strain to the smooth strain. So, this is the summary of Griffith's experiment. So, he found that live S strain bacteria in dead mice, suggesting that the harmless R strain has transformed into the S strain by acquiring some transforming principle or some molecule that has resulted in this transformation. So, he concluded that a heritable substance is responsible for this transformation. So, that was for the first time that he was able to show that there is some molecule which is responsible for this hereditary effect that we see, okay. And the experiment is explained here that I have just mentioned. Now, we know that, okay, it is most probably DNA, RNA or protein, one of these are responsible. So, to do that, several experiments were done. One of the most important experiments was done by Avery, MacLeod and McCarthy. So, it was done in 1944. So, they built upon Griffith's finding and provided strong evidence that it is DNA which is the genetic material, right. So, this research was done in Rockefeller Institute for Medical Research, which is in New York City. So, they tried to purify the transforming principle which is responsible for this transformation of the rough strain to the smooth strain. So, they isolated different macromolecules, DNA, RNA and proteins from the heat killed S strain bacteria, okay. So, then they performed selective enzyme treatment. So, what they were doing, if you add proteinase, it will destroy the proteins, right. So, only the RNA and DNA will remain, and they saw that transformation happened. So it means that protein is not responsible. When they added RNAs, it destroys the RNA, which means that protein and DNA remained, transformation happened. So again, RNA is not responsible. When they added DNAs, which destroys DNA, no transformation happened. So there is no DNA, but there is protein and RNA, but no transformation happened, which means that if you destroy DNA, you cannot get this transformation. So it is the DNA which is responsible for this transformation. This was further proved by another classic experiment done by Hershey and Chase. So this is classically known as the Hershey-Chase experiment. So this was done by Alfred Hershey here and Martha Chase in 1952. So this also provided definitive proof that it is the DNA which is the genetic material and not proteins. Okay. Later it was shown that RNA can also be a genetic material in case of bacteria. So what Hershey and Chase did was they used T2 bacteriophage. So this is a virus which infects bacteria, and the way this virus infects bacteria is it will sit on top of the bacteria and inject its DNA which is present inside. So the virus will not enter, it will only inject the DNA, and then that DNA will go inside the bacteria. From that, protein and other things will be synthesized, then the whole virus will be assembled and these viruses will bud out from the bacteria by rupturing the bacteria, right. So bacteriophage virus can infect bacteria by injecting its DNA, and this virus is made up of protein. So this coat, the envelope that is present, that is made up of protein. So they used radioactive labeling to track which molecule entered the bacteria during infection. So which molecule is actually resulting in the infection of the bacteria? Is it the protein of the virus or the DNA of the virus, right? So at that time it was not known which is going in. So the DNA was labeled with phosphorus 32 and sulfur was labeled with sulfur 35. So both are radioactive. Now it is important to note that DNA contains phosphorus. Normally if proteins are not phosphorylated because of post-translational modification, then you will not have phosphorus in protein, right? So phosphorus will be only in DNA and sulfur will only be in protein because we have methionine, we have cysteine. So there is sulfur in the protein, there is no sulfur in DNA. So we can distinguish between DNA and protein based on these two radio labeled atoms. So the experiment was that they labeled the phages, right, and they infected the E. coli, which is the bacteria, and the mixture was blended. So again it is not very harsh blending. The only way it was sort of mild blending, so that the virus that is sitting on top of the DNA, top of the bacteria will fall off, right. So you will separate the virus from the bacteria, and then if you spin down, the bacteria which is heavy will form a pellet at the bottom and the virus will be in the supernatant at the top. The result that they found that in the bacteria, DNA was found inside the bacterial cell, but the supernatant had the protein. Okay, so there was P32 radioactivity inside the bacteria. There was no S35 radioactivity. The S35 radioactivity was only in the supernatant, right, which means that protein did not enter, it is the virus which entered, and it is the DNA which entered the bacteria, and this DNA is responsible for producing new viral particles inside the bacteria. So this is the schematic of the experiment. So we are growing this bacteria with two different labeled viruses. In one case sulfur is labeled, in the other case phosphorus is labeled, right, and then you infect the bacteria with the virus. So the virus attaches on top of the bacteria like this, right, and then it will inject the DNA. So this is something that we know now, but it was not very clear at that time, right. So to see what goes inside the bacteria, is it the protein or the DNA, they blended this. So what they did was, it will just detach the virus from the bacterial surface, right. So then you spin it down in a centrifuge. So the bacteria is heavy, it will form a small pellet at the bottom, but the virus, since it is so small, it will be in the supernatant. So it will still float in the water. So if you separate the supernatant and the pellet and you check for radioactivity, for S35 labeling, they found all the radioactivity in the supernatant, nothing in the pellet, which means that the protein did not enter the bacteria. But when they used P32 labeling, they found that all the P32 was in the pellet, there was nothing in the supernatant, right? Which means that the DNA is the one which entered the bacteria, not the protein. So this again conclusively proved that DNA is the genetic material, it is the transformative principle. So during that time, while all of these experiments were being done, people were also thinking about, so it was more or less clear that DNA is the genetic material, but it was not clear how it looks. So how it stores information and how it passes on information, how the information is copied. So these were all very important questions, and to answer that, it was very important to determine the structure of the DNA. So there was a big competition that was going on to determine the DNA structure between various labs. Two prominent labs were, one was led by Linus Pauling, so he was in Stanford at that time in USA, and he published the first structure in 1953, which was this triple helix model, right? So where the phosphate groups are inside and the bases are outside like this, and it turned out to be a wrong model. But then this model was something that also led to the publication of their model by Watson and Crick in the same year 1953 later on, and we will see that that was the correct structure. So even though this was not the correct model, he was able to influence this sort of race for discovery of or explanation of the DNA structure. So Rosalind Franklin, she had worked a lot on DNA fibers, not crystals, fibers, and she had collected a lot of very beautiful X-ray pictures like this, right? So these are the diffraction spectrum of X-ray. This one is famously called the photo 51, and this type of pattern was something that eventually led Watson and Crick, specifically Crick, to believe that DNA forms some sort of helical structure. So that formed the basis of their model, and they were able to show that DNA form, the two strands of DNA are intertwined together and they form this double helix structure. Also, certain pitches that you can see here that also told them that DNA has a uniform width and it has grooves in the helical structure. So all of these were important clues in determining the final structure of the DNA. So using this data from Franklin and Chargraph's base pairing rule, where AT and GC were present in a certain ratio, Watson and Crick were able to describe the accurate double helix model of DNA in 1953. So this was published in their Nature paper in 1953. So their key findings, as we have already seen, that there are two antiparallel strands. It has right-handed double helix. It has two grooves, major groove and minor groove. A always pairs with T, G always pairs with C. The sugar phosphate backbone is on the outside and the bases are on the inside, right? So once this structure was published, so this is the famous double helix structure, it was very clear to everyone that this has to be the right structure because it explained a lot of things, right? So this, it is important to note that this double helix structure was actually drawn by Francis Crick's wife, Odile. Okay, so there is a very interesting paragraph. So this was a very small paper, you can actually go and see it, and there is this very interesting paragraph which I have reproduced here. So it says that it has not escaped our notice that the specific base pairing, so this base pairing in the double helix structure that we have postulated immediately suggests a possible copying mechanism for the genetic material. So since you have two strands which are complementary to each other, and that was the basis of this double helix structure, you can imagine that if you open up the two strands like this, you can use one strand as a template to copy the other strand. So this copying mechanism became obvious because of this double helix structure. So it actually provided a huge impetus in the field of molecular biology at that time. So based on that, then as we have already discussed how DNA replication happens, so all of those things were discovered. So 1953, the DNA double helix structure was published, and within 3 years, Francis Crick also published this hypothesis, which he pronounced as the central dogma of molecular biology. So once information has got into a protein, it can't get out again. Information here means the sequence of the amino acid residue or other sequences related to it. This is we may be able to have. So what he proposed that DNA has information which can be copied to make more DNA. That information can be used to make RNA. Information from RNA can be used to make DNA. Information from RNA can be used to make more RNA. Information from DNA can be used directly to make protein, or information from RNA can be used to make protein, right? But never protein can be copied into protein, protein can be copied into RNA, and protein can be copied into DNA, right? And so far, this has stood the test of time. So this was proposed in 1956, almost 75 years back, and still this has stood the test of time. So all of these steps have been discovered now. So this is the modern version of the central dogma of molecular biology, and it looks exactly what Crick had predicted 75 years back. So one important step in DNA replication is the semi-conservative conservative replication of DNA, right? So Watson and Crick, the structure showed that okay, you can separate the two strands and you can copy it, right? But what was not known was if there are two strands and if they are copied into two new strands, the two original strands, do they remain in the original parent cell, and the two new synthesized strands, do they go to the daughter cell, or both the cells inherit a hybrid strand where one copy is original and the other copy is newly constructed, right? So that is semi-conservation of DNA replication, and that was proved by Messelson and Stahl by their famous experiment. Again, this has been described, so I will briefly go through it. So in 1958, they showed this using bacteria, replication of bacteria and nitrogen isotopes. So there are two isotopes of nitrogen that they used, N14 and N15. So N15 is heavier than N14. Bacterial culture in a media containing these heavy isotope of nitrogen 15 were grown for several generations, right? So after several generations, bacteria were transferred to a medium containing the lighter nitrogen N14. So if you grow it for several generations, all your DNA bases will have N15, right? Now if I transfer it to N14 media and let it grow once, so there is only one replication, I will know whether I am getting N14 incorporated in some cells or I am getting mixed hybrid of N14 and N15 in all the cells. So how do you do that? They tested that by using ultracentrifugation, because the density of both strands as N15 will be different from both strands as N14, which will be different from one strand as N15 and one strand as N14, right? So they can actually separate out these three types of strands, and that is exactly what they did using a cesium chloride gradient density. So first generation, both were double stranded, and so initially both were double stranded. In the first generation, they found this intermediate band which was a mixture of 15N and 14N, and then from second generation onwards they found a new band where they found both strands were N14, but they never found both strands with N15, right? And that is shown here. So this has been explained in details in the previous lecture, so I am not going to go through this too much. But what they found, that the original double strands where both were N15 labeled never again showed up. In the first generation they found this hybrid, and then from the second generation onwards they found both were N14 and the hybrid N15 N14 started decreasing, right? So this showed that DNA replication happens in a semi-conservative manner. So here is a summary of all the major discoveries, right? Watson and Crick showed the double helix structure in 1953. Semi-conservative replication was shown in 1958 by Messelson and Stahl, and then Arthur Kornberg worked a lot on the DNA polymerase, and he explained how DNA replication happens or is catalyzed by DNA polymerase. DNA to RNA transcription was shown by his son Roger Kornberg. RNA to DNA, the reverse transcription, was shown by David Baltimore and Howard Temim, right? And then RNA-dependent RNA polymerase discovery was also done, and then I will discuss about this last step, which is the translation. So before that, some more important discoveries were made. One of them is the one gene, one enzyme experiment. So one gene codes for only one enzyme, and that was shown by Biddle and Tatum. So this is famously called the one gene, one enzyme experiment. So they were present in Stanford, and what they did was they irradiated X-rays on the haploid strains of the fungus in crassa, right? So if you take the wild type fungus and if you take this irradiated fungi, right, and then you cross them, what they found was that they were able to produce mutants. So they were selecting for these mutants, so they can find mutants which had lost their ability to synthesize the amino acid arginine, which means that if you want to grow this mutant, you have to supply arginine in the growth medium. Okay, so you have to supply arginine in growth medium and only it will grow, otherwise it will not grow. So that way you can select for this type of mutants. Now crosses between these type of fungi produced 4 distinct mutant strains that were defective in different steps of arginine biosynthetic pathway. So if you think of arginine biosynthetic pathway, let's say there are 4 enzymes which are important, and they found that there were 4 different mutants which was defective in arginine production, which means that these mutations were in one of these 4 enzymes, right? So that is why you have 4 different types of mutants. So each mutant lacked a specific gene that encoded one of the 4 enzymes that participated in the production of the arginine. So that meant that one gene is producing only one enzyme, because if one gene produced a defect in multiple enzymes, then you will see much more defects in those mutants, right? Now, based on all of this information that was being produced at that time, Crick and Griffith, they came up with this concept of comma-less triplet based non-degenerate code, that there has to be some code by using which this information that is stored in DNA and RNA is getting converted into protein. So in 1957, Francis Crick, John Griffith and Orgel, they published a paper where they proposed their scheme of non-degenerate code under which each amino acid was encoded by only a single codon. So this was eventually proved to be wrong, because now we know that multiple codons can code for a single amino acid. So there is degeneracy. So, but two important things that came out from their paper was that there are these codons which comprise of 3 nucleotides, which proved to be right, and the code is probably non-overlapping, which means that 3 nucleotides will code for one amino acid, then the next 3 nucleotide will code for the next amino acid, so on and so forth. So these codons will not overlap like this, okay, one on top of the other. That is not permissible. And eventually this was proved by Conrad's experiment on RNA genome of tobacco mosaic virus. So Heinz Conrad and Gerhard Schramm, so they worked on tobacco mosaic virus, which has an RNA genome, right? So their experiment in 1956 was groundbreaking in 2 ways, because it showed that RNA carries genetic information. So that was a big discovery. And the second outcome of their experiment was that these codons, they are non-overlapping, which was proposed by Crick and others in their paper. So in this case, they produced single nucleotide mutants of the RNA genome of the tobacco mosaic virus. The mutated RNA was combined with normal tobacco mosaic protein and used to infect tobacco plants, right? So in this case, the resulting virus retained the mutation, proving that the genetic change was passed down through RNA, not protein and not DNA. So they were able to mutate, and then that mutation was passed on through several generations. So it proved that RNA is the genetic material. And also the point mutation in the RNA genome of TMV resulted in substitution of a single amino acid. So if there is a mutation, then it is present only in one codon and it affects only one amino acid. So it means that there is no overlapping of codon, which means that single mutation is not affecting multiple amino acids. And so since that is not happening, it means that these codons are non-overlapping. So this non-overlapping nature of codon was also established by this set of experiments that was done by Conrad and Scram. So the experiment is briefly shown here. So you have this TMV RNA, okay. So it was treated with nitrous acid HNO2 and the viral reconstitution was done. So you have the mutant RNA, viral protein. So you have the reconstituted virus which infects the tobacco leaves, right, and then they will replicate. So now you isolate this viral protein and then you do this nucleic acid sequencing. So what they found that in the newly produced viruses the mutation is present, which means that since we are mutating RNA and it gets carried over to the protein, so RNA is carrying the genetic information and only one amino acid was mutated. So if you produce a single nucleotide mutation, only one amino acid is mutated, which means that there is no overlap of the codons. So RNA dependent RNA polymerase, RNA as a genetic material was established by them, and also non-overlapping codons for RNA. Again around that time, Crick produced another hypothesis. So this is something that is very important to note, that to do good science you should have a hypothesis, otherwise you cannot work on something. So what I am trying to say is that one has to have some hypothesis that explains a certain observation, and then you can design really clever experiments to test that hypothesis. So Crick is called as a theoretical biologist because he produced this really useful hypothesis that drove all the experiments of all the important experiments of molecular biology at that time. So again, another important hypothesis that he came up with was that there must be an adapter molecule that links the genetic code in RNA to amino acids. So these 3 codon should get converted to one amino acid. So there has to be some adapter molecule which will convert this or do this translation, right? And he said that there has to be some adapter molecule. And 10 years later, Robert Hawley, he isolated the first tRNA molecule. It was coding for alanine. From yeast, he determined its structure and proved that this adapter hypothesis is correct. And in the RNA, in the tRNA or the transfer RNA, the anticodon was found which is complementary to the codon that will be present on the mRNA. So then Zeminick and Hoagland, so effectively by using in vitro protein synthesis system, these two scientists, they identified and characterized the tRNA and amino acid activating enzyme. So there has to be a tRNA, but then if once that is used, the tRNA will go back and the amino acid has to be recharged on that empty tRNA. So you need activating enzymes for that. And these were discovered by Zeminick and Hoagland. So they prepared cell extracts from rat liver containing ribosomes and other protein synthesizing components. So they added radioactive labeled amino acids, for example, carbon 14 leucine. Then they observed that before the amino acids were incorporated into proteins, they first bound to a small heat stable RNA molecule, which was the tRNA, and that is how they demonstrated that amino acids must first attach to tRNA before being used in protein synthesis. So these were all very important steps that are present in protein synthesis, which we now take for granted, but they were all discovered by these really carefully designed experiments by different biochemists. So these also confirmed Crick's adapter hypothesis. Okay, so now there is this adapter, but you have to discover the codon table, that which 3 triplet of nucleotide code for which amino acid. Now remember that Crick had originally proposed that the genetic code will be non-degenerate. So only one codon will code for one amino acid. But later when people tried to look into that, they found that this is not entirely correct. So Nirenberg, he used this cell-free system that was discovered, I described in the previous slide, to see what type of RNA is producing which type of protein. So he used poly-U RNA, right? So all the nucleotides are uracil, and he found that the protein that is made up of entirely of phenylalanine. So it means that UUU codes for phenylalanine, right? So similarly, when he used poly-C, polycytosine, so all the nucleotides are C, he found that it produces polyproline. So this way, he was able to decode 54 of the amino acid codons by 1966. Parallelly, another scientist, Hargobind Khorana, he was working in Harvard on the same problem, and he also used different types of synthesized RNA molecules to see what type of amino acids are getting synthesized. So he was able to confirm that AUG is the start codon, and there are 3 stop codons which are UAA, UAG and UGA. So together Khorana and Nirenberg, they were able to produce and show that there are actually multiple codons which can code for certain amino acids, and they built the whole genetic codon table which looks like this. And this data was also curated by Francis Crick. So they would send their data and Crick would tabulate all of them in a form like this to prepare the genetic code table. So the 1968 Nobel Prize was awarded to Robert Hawley, Hargobind Khorana and Marshall Nirenberg for their interpretation of the genetic code and its function in protein synthesis. So now we know that this part required a work from a lot of scientists, and their names are listed here. So what we show as just one slide is actually contribution of some really smart people working in tandem and figuring out all these different details by doing some really clever experiments. So one more topic that is present in this, which is RNA splicing. So it was discovered in 1970s by Richard Roberts and Philip Sharp, so who later received the Nobel Prize in 1993 for their work. So what they showed, so they were studying adenovirus, and using electron microscopy, they were hybridizing the messenger RNA and the DNA. So RNA is synthesized from DNA, so they were hybridizing this messenger RNA with the DNA, and they found that it was not a perfect match because there were a lot of regions in the DNA which was not matching with the RNA, and they will form these large loops like this. So in red you have the messenger RNA, and in blue you have the DNA. The DNA codes for the same information which is transcribed into the messenger RNA, but when you hybrid them, you see that the DNA comes like this, then there is this big loop because there is nothing that matches it, and then it matches, so it forms like this, a double strand, and then there is another loop, and then again it forms the complementary strand, then there is another loop, so on and so forth. So these loops are what they call so these are the 2 regions that they called as the introns and the exons, right? And they hypothesized that it is these introns which are spliced out, and the exons are joined together to form the mature messenger RNA, right? So this is your mRNA splicing. And then later on, Tom Che, he showed that this mRNA splicing can be catalyzed by the messenger RNA itself, the RNA itself. So where he showed that RNA can also have catalytic activity, and he shared the Nobel Prize with them in 1993. So in conclusion, landmark experiments in molecular biology have fundamentally shaped our understanding of life, which is the molecules of life, right? So from the discovery of DNA as genetic material to the double helix, all of these things have paved the way of all the modern molecular biology and genetics that we do today. So they form the fundamental basis. So we have seen how Nirenberg and Khorana, they have discovered the genetic codon, right? So all of these things that we have seen in this lecture. Discovery of RNA splicing by Roberts and Sharp. So together, these breakthroughs have provided a comprehensive view of molecular biology from genetic information storage to its expression as the functional protein, which do all the work in our body. So these foundational discoveries continue to drive advancement in biotechnology, medicine and genetic engineering that we do today. So, if you are interested in reading about these landmark experiments, you can follow this book called Landmark Experiments in Molecular Biology by Michael Fry, and then these all other books are also very important and you can use them as references. Thank you.